Good afternoon. This session is about the transplantation. So basically, transplantation is the technique of transferring. The word meaning itself is transplantation means taking from one place and shifting to another one. So transplantation is the implantation of non-self tissue into the body, and it is a technique of transferring cells, tissues or organs from one side to another. If you have any deformed organ in your body or deformed tissue in your body, that is transplanted, and we all are familiar with the common transplantation is the kidney transplantation. That's a major one, but still that's a common transplantation which is heard by people. Kidney transplantation. If a person is having a defective kidney which cannot be cured by medication or anything, he have to get a functional kidney from another individual. That's known as transplantation. So it is a technique of transferring cells or tissues or organs. It can be cell, like if you have this leukemia or blood cancer like that, cells are transferred, tissues are transferred, or even organs like kidney, liver, or heart. This is known as transplantation. And it is actually done to control the fatal conditions among the human beings. And the process of this process of taking is called the transplantation. And the organ which is transplanted is called a graft. Graft is a or a transplant. It can be tissue or organ from one part of the individual. For example, if you are transplanting a kidney, in that case the graft is a kidney. So the tissue or cell which is transplanted is called a graft. And the individual who donate this organ or graft is called a donor, and the individual who receives this organ or the transplant or the graft is called the recipient. So that is transplantation. Graft is the tissue, organ or cell which is transplanted. Donor, one who provides it. Recipient, one who receives it. So this transplantation have a very history. The history is very long back itself. People have started the transplantation that early from the second century BC itself by Sushruta and all. They have done this transplantation with the skin, and this transplantation has got a very wide application during the World War time. During World Wars like World War One and Two, lot many people got their skins burned by this fire and all. And those people who were having this burned skin underwent this transplantation of skin. So it is very, very useful in case of during the time of World War II, where when the skin grafting was done. And Sushruta in the early second century has also grafted skin for nose reconstruction. If you have these reconstruction of your nose and the skin is the transplantation was done from the early time of Sushruta itself. But a very successful transplantation was actually reported by Alexei Carrel in 1908. Alexei Carrel was done a systematic study of this transplantation, and the successful transplant was done with the help of that is was done in kidneys. That is, the kidney was transplanted in a series of cats, and few cats survived for almost 1 month. So that was a successful transplantation that was done by Alexei Carrel in 1904. Sorry, 1908. But after later on, he continued his studies in transplantation, and he was awarded Nobel Prize for in medicine in 1912 for his work on vascular suture and the transplantation of blood vessels and organs. I said the transplantation of blood vessels and skin is very widely accepted by the human community because during this World War time. Then the major, then after that, so many people like has worked on this transplantation on organs like liver, heart, kidneys, etc. and pancreas, bone marrow, cornea. There are different types of transplantation. And corneal graft is the transplantation in which the grafts is taken, that is the cornea is transplanted, that is known as corneal graft. I said the common one is kidney transplantation, and the successful kidney transplantation was done in 1954 by Peter Bent. Then heart transplantation was done in 1967. In 1967, cardiac transplants was done successfully in a 40-year-old patient. Then liver transplantation, then bone marrow transplantation is done in case of people who are having this blood cancer. Those people, the blood bone marrow transplantation is done in case of this leukemia or the blood cancer, because they do not have the blast cells which produce the RBC and the WBC. So these are the different organs and tissues which are transplanted: cornea, heart, kidney, then liver, bone marrow, etc. and cornea. So that is why in eye donation, you are very familiar also. In eye donation, the cornea is taken from a person who is dead, and the kidney and all will be taken from those people who have met with a severe accident, or whose brain death has occurred or something like that. Then let us see the different types of transplant, or the types of different types of graft. So the different types of grafts are, the classification of the transplant or the graft is done based on the individual from which the graft is taken. And in this picture you can see a mice and you can see a cat. So from a cat, you can see if a transplant, or if a organ of a part of skin or blood vessel is taken, is taken from one part of this mice, and it is transplanted or attached to another part of the same mice itself. That is called autograft. Auto means self. Auto means self. So autograft is if a tissue or part of body or part of tissue like skin or blood vessel is taken and it is attached on another part of the same organism, that is called the autograft. And this is actually frequently done in people like in bypass surgery. The blood vessel will be taken from your thigh region, and skin will be taken from other parts if to be transplanted onto face in acid burns like that. That is known as autograft. One part of the blood vessel or skin will be taken from one part of the body and it is fixed on another part of the same organism. See here, one part of the mice to another part of the mice itself. That is known as autograft. This is known as autograft, from mice to the same mice itself. Now, next is isograft. Isograft is also called syngeneic graft, and it is the tissue is transferred from the donor. Tissue is transferred from the donor to a genetically identical recipient. It is transplanted from one part of the from the donor to a genetically identical, means it is between the identical twins. Here this is known as iso, means same, genetically same. Genetically same or identical sibling or clone. To the clones, or genetically same or identical twins, if the transplantation is done, that is known as isograft. Then next is allograft. Allograft means if the graft transplantation is done between two organisms which are not genetically same. That means, see here, between, if the transplant is between a white mice and a black mice, they are genetically different. If the transplantation is done between two genetically different mice, that is called allograft. Allograft means genetically different but same species only. Genetically may be different, but same species, that is between two mice. That is known as allograft. Genetically they are different, but same species. And that is allograft. The next is xenograft. Xenograft means xenograft is between species. See, here a graft is taken from mice to a cat. That is known as xenograft, between two species. So, the grafts are of four different types. Autograft, between the same organism. From one part of this organism to another part is autograft. Isograft, between genetically similar organisms. Allograft, between same species but genetically different. Xenograft means different species. These are the different types of graft. You can see here, that is autograft. I said autograft means from one part of the body to another of the same organism. So the antigen present in autograft is same as that present in the body. So the autograft is less risky when compared to other grafts because it is from one part of the organism to the another part of the same organism only. So the antigen which is present on the autograft is same as that is present in the body. So it will not cause any serious rejection in the rejection. So the immune system will recognize the autograft as its own. So the rejection is null or very, very less. So no immune response is elicited. So autograft survive throughout the life. That is why bypass surgery, when it is being done, the blood vessel is taken from your thigh region and it is then transplanted to your heart region. That is bypass. So when after the bypass surgery, the people will live long without any problem because it is from the same organism itself. So the immune response will not immune system will not work against it. So transferring the healthy skin to burned area, as I said, in acid burns or burns on the face, and all skin will be taken from some other part of the body like hand or leg and it is transplanted on the face. Then use of healthy blood vessels to replace the coronary arteries, that is the bypass surgery. So autograft is always the best one in case of the skin and this artery, uh, this bypass surgery and all, because no immune response. The next is isograft. Isograft is also a syngraft. Isograft means between genetically identical individual of same species or between the twins and all. Isograft between the members of the identical twins. So the tissue is transferred between genetically identical individuals. They are genetically almost identical, but in isograft the histocompatibility antigens are identical. So the graft is not rejected. So, but the chance of getting is very less. Between the identical twins, it is the graft rejection is very less because the identical twins will have almost same immune system. But it is not possible in all cases, all times. Example, human isograft can be performed between two twins. They have the same genetic, that too between the identical twins because they develop from the same ovum, that is identical twins. Allograft between, I said allograft between same species but two genetically different members. Maybe a black mice and a white mice, or a yellow monkey and a black monkey, like that. So tissue is transferred between two identical species. So here the antigens are dissimilar. Here allograft, the antigens are dissimilar, so the immune response is will work and the graft rejection is very high. The chance of the graft to be rejected is very high because in human beings, you might have heard, if you receive a kidney from a person a person living in Kerala, receiving a kidney from a person who is living in London, I mean, they are genetically they are the same species itself, but they are genetically different. So the chance of graft rejection is very, very high. So in humans, graft is transferred from one individual to another. Kidney transplantation also you might have seen in the newspaper or so. We kidney like that. Any person can donate kidney, but the chance of rejection is very high when compared to the identical twins. The next is xenograft. I said xenograft is transplant between species and the graft rejection chances extremely high, very high because two different species will have two different immune system and also they have a very the cross reaction will be very high. That means transplantation between a human and a human being receiving a kidney from a monkey or human being receiving a heart from a monkey or receiving a heart a organ from a pig to humans, etc. And this is called xenograft and the chance of rejection is hyperacute rejection means the rejection is very high. Now, even though after transplantation, what happens is the body have the transplantation rejection after a transplantation surgery. The body has to consider the transplanted part as its own. It should not reject it. So, transplant rejection occurs when the immune system of the recipient of a transplant attacks the transplanted organs. If you are taking a kidney from a pig into a man's body, a human being's body, the being's body will reject it because it is from a different organism. So the immune mechanism that is used for the rejection is called the immune response. The ability of this immune system to distinguish between the self and non-self we have learned in previous classes is called the tolerance and all. So the major transplanted antigens are of the rejection of the transplant is of blood groups, ABO blood groups and human leukocyte antigens system. I said that is the human leukocyte antigen system or HLA and the blood group. First of all, when a person is between two individuals, if a human being is planning to receive this a transplant from another individual, the first step is you have to check for blood group compatibility. If it is two, they are having two blood groups that will result in blood group incompatibility. Then second step that is the human leukocyte antigen. If there is a mismatching between the major histocompatibility complex or MHC, you have learned that in previous classes, MHC. The MHC matching mismatch will greatly result in graft rejection. So, major histocompatibility complex should match between the donor and the recipient, and that will result in the graft rejection. And this is what are the precautions that can be taken against this graft rejection. Of course, first one I said the blood group or the ABO blood group system. So there should be the blood group should be matching between the donor and the recipient. Donor one who gives, recipient one who receives. Both of them should have the same blood group. So ABO system is present should be ABO matching should be there. Otherwise, the graft rejection will be very high. Then, cross match test. That is to determine the presence of the HLA antigens. The previous slide we told HLA antigen of human leukocyte antigen. And this cross match test is used to determine the presence of the antibodies to HLA antigens. And then only that person will go for the receiving the graft from a donor. Now let's see the mechanisms of graft rejection. The graft rejection, the degree of immune system or immune response to a graft based on the type of graft. As I said, the graft rejection is nil in case of autograft. The graft rejection is a little bit more in case of allograft, then isograft a little more, then xenograft, the graft rejection is very high. So the degree of immune response will vary from individual to individual. The degree of graft rejection varies from one individual to another individual. Sorry. In general, the skin grafts are rejected faster than any other tissues. The skin rejection is very, very high. That is why usually the skin transplantation is done with the help of women. A skin transplantation is done from one part of the body to another. So the graft rejection is of different types. One is sensitization phase. That is a mechanism. During sensitization stage, the antigen-sensitive lymphocyte from the donor graft will migrate into the lymph nodes. That is, when this antigen mixes with the immune cells in the body, it stimulates the T helper cells production or Th cell proliferation, and that will result in start the immune system will start functioning against the new organ. That is what is known as the sensitization phase. That is, the body will the immune system will start working against the graft. Then next is effector stage. Effector stage means the effector cells will be generated. That is, the Th cells will be produced and the Th cells will elicit the production of effector cells, and this effector cells will migrate towards the graft and will destroy the graft. That is known as effector stage. So, sensitization phase, a new transplant. This is a transplant or a graft. This is a transplant, and from this transplant, when the transplant is there in the body, it will elicit the production of the leukocytes. That is, the leukocytes from this or the WBC from the transplant will elicit the production of Th cells. Th cells, and the Th cells will help in the proliferation of this effector cells. Effector cells, and this effector cell will act on this transplant and will destroy it. This is known as sensitization and the effector stage. Now, the type of rejection will depend on the amount of antigen which is present. So, first happens according to the pattern of rejection or a rate of rejection. Rejection is of different types. One is hyperacute rejection, acute rejection, and chronic rejection. First is hyperacute rejection. Hyperacute rejection means the rejection will take place within minutes after the transplantation surgery. Hyperacute rejection occurs within minutes of the surgery, and it will result in the clumping or clotting of RBCs or red cells in the body, and this is very, very serious, and it will cause severe inflammatory response in the recipient's body. This is known as hyperacute. Within minutes the graft will be rejected. Next is acute rejection. Acute rejection means for initial phase it will not reject the organ. After 1 week or 2 weeks the body will reject. That's known as acute rejection. Acute rejection works through this HLA antigen or human leukocyte antigens. And the T lymphocytes play a key role. The acute rejection, the T lymphocytes play a key role in acute rejection. T lymphocytes play a key role in acute rejection, and the acute rejection takes place after 1 week or 2 weeks. Then chronic rejection. Chronic rejection occurs only maybe after 1 year or 1.5 years of transplantation. The rejection takes place, and it is actually characterized by this narrowing of the vascular cells, or the blood cells will get narrowed, and that will result in the narrow or thinning of the vascular cells, and this includes both humoral and cell mediated responses. Because it is cell mediated, the rejection is very slow. After 1.5 years only the organ will be rejected. These are the different types of rejection. So mechanism of rejection include a sensitization phase and the effector stage. Then the different types of rejection is hyperacute within minutes, rejected, acute rejection after 1.5 weeks, chronic rejection after 1 or 2 years. Now, graft versus host reaction. That is when a foreign graft is transplanted into the body, it will develop the immunocompetent cells are grafted into an allogenic recipient whose immune system is compromised. That means the immune system of the recipient has to be suppressed for the survival of the graft. So the immunocompromised person will his immune system has to be suppressed. For example, if B is the individual who is receiving the graft, his immune system has to be suppressed. That is, immunosuppressive drugs will be given. So, graft versus host reaction means the matching is done by suppressing the immune response of the recipient. The, the reason for this is the production of this. The graft cells survive. Grafted cells survive in the immunocompromised host even though his immune system is suppressed. Again, the immune cells will fight against the foreign graft. So the graft versus host reaction brings to the damage of the host cells and the host itself. That is, the recipient itself will be affected. So when the grafted tissue has mature T cells, they will attack the recipient's tissue leading to GVHR or graft versus host reaction or GVHR, and the prevention and control of graft versus host disease will help in the survival of the grafted tissue. The mechanisms of graft versus host reaction are the graft lymphocytes. The recipient's lymphocytes will aggregate on the host lymphoid organs. Then the graft lymphocytes are stimulated by the host lymphocyte. Then it is stimulated by the lymphocytes of graft produce lymphokines. Lymphokines will activate the host T cells which produce polyclonal B cell activation. That is what I said, the cell mediated. Activated B cells react with the self antigen when they destroy the transplant or the host. So, in order to prevent the rejection of the graft, certain things has to be taken into consideration for the effective survival of the transplant or the graft in the body of the recipient. And these are, that is, first step is immune suppression itself. The person who received the graft, his immune response will be suppressed by preventing will be suppressed, and this suppression is done with the help of drugs and immunosuppressive therapy, and there are several drugs which is given to these people, and one drawback of this is that a person who receives the graft will be given immunosuppressive drugs and he will be susceptible to diseases. So he has to be kept, that person has to be kept in house or he should not go into crowd and all because his immune system will be weak because we are giving him immunosuppressive drugs. So for the effective survival of graft, two things are done, that is drugs are given or immunosuppressive therapy is done. And the different types of drugs which are given are, the common drugs are corticosteroids. Corticosteroids such as methylprednisone or prednisone, prednisolone, etc. are some of the corticosteroid drugs, and these drugs are given to prevent the graft rejection. Now, next, another drug which is given is cyclosporin, and cyclosporin will suppress the cell-mediated immune reactions, whereas the humoral immunity is affected. So, different drugs are given. Cyclosporin is given to suppress the cell-mediated immune system because it blocks the transcription of cytokine genes. You might have learned that the cytokines are initially formed in this immunoresponse step. Then other drugs which are given are tacrolimus and rapamycin. And in addition to that, immunosuppressive therapy is also done. And this immunosuppressive therapy is done. The benefit of this immunosuppressive therapy is that it will suppress the graft rejection, but in total, his survival is very, very dangerous because he will be exposed to lot many diseases other than the one he was having. So these are the steps which are taken for graft survival. So this session was about what transplantation is, what are the different types of transplantation, which are the organs that are transplanted, the mechanism of graft rejection, graft versus host reaction, then the how you can improve the graft survival. So with this I conclude this session. Thank you for your listening.